Hello guys, my name is Nelson Viegas. I'm a high performance trainer, and today I want to bring a very controversial topic, but people are super interested in this subject, which is fat loss. What are the mechanisms of fat loss? So for fat loss, we have two mechanisms. We call lipolysis, that happens on a fat cell, which is the breakdown of the triglycerides and remove them out of the adipocyte, and the beta oxidation. So today I will try to make a short video. You guys know that sometimes it's not easy for me to make very simple videos because I always go deep into the biochemistry, but let's go. So okay, this cell here that I bring you today, this is a adipocyte or a fat cell. So as you can see, we store the fat in a form that we call triglycerides, or you can see in some books triacylglycerol, which is the same thing. So we have the lipid droplets, so the triglycerides, they are inside this lipid droplet and they need to come out to the blood to go to the muscle cell to be oxidized there. So as you can see, we have lots of receptors here around the membrane of the fat cell that are activated by two things: physical activity, calorie deficit, basically that, and one in the other one. So they both activate hormones or responsible hormones that are responsible for this action. So we have the adrenaline, basically, that connects to the beta 3 adrenergic receptors. Remember, we have the alpha receptors and the beta receptors, that I will explain the difference between them in a minute. So we have the beta 3 adrenergic receptors that are present on the fat cells and in your bladder. So when the adrenaline binds to its receptor, which is a G protein coupled receptor S, because it's it stimulates something called adenyl cyclase. This adenyl cyclase is responsible to convert ATP into cyclic AMP that activates protein kinase A. This protein kinase A activates pyrrolipins, which are proteins that they start breaking down the fat to remove the fat out of this lipid droplet. Also, PKA, protein kinase A, activates CGY58. So both of them, what they do is they come here and they activate this triacylglycerol, activating this enzyme, the first enzyme responsible for the breakdown of the triglyceride, which is the ATGL. Some books it can come like desmethrin or adipocyte triglyceride glycerol lipase. So they convert the triacylglycerol into deacylglycerol, and then you have another enzyme which is even more important, that it's the hormone sensitive lipase. So that is also activated by PKA. PKA activate hormone sensitive lipase. Hormone sensitive lipase come here and breaks down the second fatty acid from the molecule of triglyceride, converting to MAG, so monoacylglycerol. And then you have another enzyme here called monoglycerol lipase that breaks down the last fatty acid, so leaving 1, 2, 3 fatty acids and 1 glycerol. We have a transporter inside the cytoplasm of the fat cell called fatty acid binding protein that they remove the fatty acids to the blood. In the blood, as you can see, the fatty acids, they cannot circulate, so they need a transporter. The transporter, it's called albumin, that takes the fatty acid to the muscle cell to do the beta oxidation. So this is what happened. Then we have several factors here that can accelerate this process or stop this process. For example, like I said, adrenaline accelerates this process, glucagon accelerates this process, T3, which is a hormone that we produce in the periphery with the stimulus of T4, so T4 is converted to T3. GH also activates this process. So these are the hormones that they activate the fat loss. Then you have insulin, for example, that does the opposite. So insulin facilitates the fat storage. But remember, insulin, that's why in my latest video, I explain what is the difference between some mechanisms and some outcomes, okay? So I said there that insulin, and I'm saying now again, insulin activates lipoprotein lipase, that is responsible to pick up the fatty acid that comes from the chylomicrons and the VLDL and puts them inside the cell for them to come and do fat storage in case of excess of calories, excess of ATP. So insulin is responsible for that. So insulin in this mechanism is lipogenic. So insulin helps to store fat in this mechanism because in the brain, insulin works as a sociogenic hormone. So for example, as you can see, insulin activates lipoprotein lipase and puts the free fatty acid inside. So insulin also activates is receptor, IRS1, that activates phosphatidylinositol 3 kinase, PI3K, that activates protein kinase B, or you can see in some books also, AKT. This cascade of events of insulin, they activate something called phosphodiesterase 3. This phosphodiesterase 3, especially the phosphodiesterase 3 beta, blocks hormone sensitive lipase. Okay, what blocks this phosphodiesterase 3? Caffeine, that I explained in the video of caffeine. What also activates this, so remember, caffeine blocks, but alcohol, as you can see, ethanol here, activates. So ethanol helps you to gain fat through this mechanism, activating phosphodiesterase 3 beta. Also, we have the alpha receptors. So we have the beta receptors, beta adrenergic receptors, and you have the alpha receptors, especially the alpha 2. That's why some supplements that we take, they mimic the beta 3 adrenergic receptors. Like for example, clenbuterol is a preferential beta 2 agonist, but also acts on the beta 1 in the heart and the beta 3 on the fat tissue. That's why clenbuterol, one of the mechanisms of clenbuterol and ephedrine, is exactly to mimic the adrenaline and binds to this beta receptor to activate this cascade of fat loss. On the other hand, the alpha receptors, they block this process. So you see, alpha receptor, they block the adenosine. That's why we use yohimbine. So yohimbine antagonizes alpha 2 receptors. So it means that this one activates adenosine, so accelerates the process, and the alpha 2 that is supposed to stop this process, now they are deactivated by yohimbine. So the combination from clenbuterol or with clenbuterol and yohimbine, it's a good combination for fat loss. Okay. Another thing that happens is this butyrate, which comes from the fiber that we eat. So in the food that we eat, that's why in my programs I put lots of fiber, especially fruits, because the fiber is not digested. So it's converted into three types of fatty acids called acetate, propionate, and butyrate. So especially the butyrate, they activate two receptors in your intestine called GPCR41, GPCR43. They activate GLP-1 and neuropeptide Y. So what happens is butyrate, what they do, is they increase the satiation. So that's why you eat fiber. So fiber activates FIAF, fasted induced adipocyte factor, that blocks LPL. So LPL that is stimulated by insulin is blocked by this FIAF. So fiber, through this mechanism, it's converted to butyrate. Butyrate activates GPCR41, GPCR43, and they block FIAF. So this is the mechanism of lipolysis. On top of that, we have our lovely beta oxidation. Now this happens in the muscle. So in the muscle, as I explained in the video of L-carnitine, so the fatty acid that is in the blood, it comes through inside the mitochondria, first inside the cell and then inside the mitochondria, through a transporter called cluster differentiation 36. So this CD36 goes inside, and here by the fatty acid synthase receives acyl-CoA. So it starts changes the name from fatty acid to fatty acyl-CoA. When this fatty acyl-CoA passes through carnitine palmitoyltransferase 1, the acyl-CoA, so the coenzyme is removed and it's added to the molecule carnitine. And now the name of this molecule is called acylcarnitine. This acylcarnitine passes through CPT1 and CPT2. On the CPT2, removes the carnitine, as you can see the carnitine goes back again, and receives again the CoA. So the limiting factor is the coenzyme A and not the L-carnitine. When this acyl-CoA goes inside CPT1, CPT2, it comes here, it is converted again in acyl-CoA. So acyl-CoA, acylcarnitine, go back again to fatty acyl-CoA, and then goes to the process of we call beta oxidation, which is 6 stages, to be converted to acetyl-CoA, and this acetyl-CoA is the one that goes inside the Krebs cycle. We also have the PPARs that I don't want to go into deep into this part because it's very complicated. So this is also transcription factors that they increase the lipogenesis or the beta oxidation inside the nucleus of the cell. On top of that, we have our Krebs cycle that I explained last time. So the Krebs cycle is after that. So lipolysis, beta oxidation goes to the Krebs cycle. So we have here the glucose or the process of glycolysis that they break down everything into pyruvate, and you have the beta oxidation from fatty acid that they go back at acetyl-CoA. So pyruvate, acetyl-CoA, but in the end what we have is always acetyl-CoA. We have 8 stages here. We have our famous isocitrate dehydrogenase here that's supposed to convert isocitrate into alpha-ketoglutarate, but it's blocked. So it goes back again to citrate, and this citrate can go out of the cell to form triglycerides. And we have the electrotransport chain. This is a very complex part of the cell. This is the end. So like I said, in this process here, so if you go back, we can see that in a Krebs cycle, we have the FAD and we have the NAD. What they do is they remove all the ions of hydrogen, the H+, and they send it here. So as you can see, the NAD comes from the complex 1 and the FAD comes to the complex 2. So they throw all these ions of hydrogen to the top, to this called intermembrane space here. And here we have our famous antioxidant enzymes called superoxidismutase, catalase, glutathione peroxidase, thioperoxidase. So we have all our antioxidant enzymes here. So when the ions of hydrogen that pass through these complexes, in the end, they have our famous ATP synthase. So when these ions of hydrogen they come through the ATP synthase, as you can see, the shape of this, it spins and it produces ATP. You can produce from palmitic acid, which is fatty acids, until 129 ATPs, and from glucose until 38 ATPs. Here, again, we have the function of or the production of oxidants, we call reactive oxygen species. So you have the superoxyl radical anion, the hydrogen peroxide, the hydroxyl radical, and the peroxynitrite. This is the reactive oxygen species that we produce there. And then here, by these enzymes, catalase, superoxidismutase, thyroredoxine peroxidase, glutathione peroxidase, and glutathione reductase, they convert into water. So also we have foods that activate this transcription factor here called NRF2 that goes to the cell nucleus and activates this enzyme. So these enzymes are super important because they are the ones that excrete the reactive oxygen species, okay? So as you can see, all these reactions, so the oxygen goes to peroxynitrite and goes to H2O, which is water. So this is how we remove these antioxidants out of our electrotransport chain. Okay, so guys, I will stop here. This is too complicated to go now to the younger mechanisms. I can do this in another video. I hope you guys understand what is the difference now between the lipolysis, the beta oxidation, the Krebs cycle, and the electrotransport chain. So for you to understand what supplements they work there, like I show you in some videos that I did before, for example, we have here the complex 1, 2, 3, 4, the ATP synthase, but also we have here the UCPs. Remember I spoke with you guys about the UCP, so this is responsible to produce heat, okay? So if the energy is here, the ions of hydrogen is here, and they all go through ATP synthase, they will produce lots of ATP, lots of energy, but there is ways and supplements that they work on this UCP. So we produce heat, so all the ions of hydrogen, they are not taken to the ATP synthase, they also go to the UCPs. Okay guys, thank you so much for watching this video. Hope you guys now you understand the difference between all these processes, and please keep following me, share these videos and watch it over and over again for you to understand, because these images are very important, because sometimes when I explain it's complicated. I understand, because for me, I see the images in my brain, but you guys don't. So when you look at these images, you understand. I will recap the images just for you guys to see the images, okay? So we have here, I'm going out of the screen. So lipolysis, beta oxidation, Krebs cycle, electrotransport chain. Thank you guys, and see you next time.